And welcome to the Human Genetics and Genomics Seminar Series.
Distinguished speakers.
The distinguished speaker is he has a certain age to be a distinguished speaker. It's my pleasure to introduce Dr. Alex McKenzie. Alex came from Ottawa to talk to us today. Alex he did his undergraduate studies at the University of Ottawa, and then later received an MBA and PhD from the University of Toronto. He is currently the principal investigator for the Apoptosis Research Centre at the Children's Hospital of Eastern Ontario. He's a physician in the Division of Endocrinology and a professor in the Department of Pediatrics at the University of Ottawa. He's had lots of jobs during his life. He was a former CEO and scientific director of the Children's Hospital of Eastern Ontario. He was vice president of research for Genome Canada for a year. And he's co-chaired many scientific advisory committees, won numerous awards, but he asked me not to talk about the awards because it makes him squeamish and feel old. So,
thank you very much, Chris.
Yeah.
That was your secret.
Yeah.
The last award being, as Chris pointed out, 1982. Oops, I'm dragging my, it peeled off. Thanks, Chris. It's great to be here. This is my son Duncan is a very happy master's student here in immunology, and I'm very happy to be here at Dalhousie. And seeing old friends like George Robertson as well is a wonderful thing. So they told me I had 40 minutes last night, but I got 2 hours. Don't worry, I got this covered. So basically, I'm going to talk about a lot of stuff that people may already know as far as the next-gen sequencing, rare disease identification thing, and then in the last bit of my talk, we'll talk about how it applies to spinal muscular atrophy. So this is a mantra that's becoming increasingly familiar to many of you, the idea that of the 23 or so thousand genes that make up the human genome, roughly a third of them, when mutated, will cause a rare genetic disorder. A question is, what is happening to the other 2/3? Why do mutations therein not cause disease? And I don't know the answer. If anybody's smart enough to come up with the answer to that, do let me know. The other thing about rare diseases is they aren't rare. About 1 in 15 Canadian families is affected by a rare disease. About half of them are cloned at this stage, roughly 3,000, 3,500, with the other remainder uncloned. But that's changing rapidly, as I think most of you know. In the last 5 years, the advent of next generation DNA sequencing, the rather remarkable drop in cost. This is the Moore's law, in this case halving at a certain period of time. This is the actual drop of the cost of the sequencing of genome, coming down from 100 million back in the original sequencing of Jim Watson's genome and others, to now where it's getting approximately $3,000 for a genome, so a dramatic drop. And so it means that Huntington's disease was originally mapped in '83 by Jim Musella. It was ultimately cloned in 1993, it took a decade for that to happen, whereas my close colleague Kim Boycott, heading up the FORGE consortium with many of you in the room, I think the last count was 100 disease genes identified over a 2-year space, roughly every 2 years, every 2 weeks, every week to 2 weeks there's a novel gene discovered. Her office is right next to mine, having spent about 12 years identifying the wrong gene for spinal muscular atrophy. It gets a bit annoying when you're, oh, another gene. Congratulations, Kim. I try to weasel my way into the authorship list every now and then, it doesn't happen. At any rate, but it is all joking aside, a very exciting period, because in essence, over the next 5 to 10 years, a third of all human genes, when mutated, will be known. It's a remarkable wealth of pathogenic knowledge. It'll profoundly affect our understanding of how we are strung together at a molecular level. Textbooks and monographs will all be rewritten. And in addition to these increasingly ultra-rare, as we move on to cloning the remaining thousands, we're getting down to 17 in the world, etc., really ultra-rare. But with these clonings will come insight into common disorders as well. There's also a human element to this. Right now, there's this idea of diagnostic odyssey for those with rare diseases. Children, and they're mostly children, but children and adults with rare diseases and the affected families are really the first responders, along with the cancer community, to the next-gen sequencing, and they're the ones experiencing the most manifest benefit, I think. Currently, and I think people, geneticists in the room know this, that basically there can be a long wait for the diagnosis, and when the diagnosis comes, as much as half will be incorrect, and maybe 50% of the remainder will never ultimately receive a diagnosis. The Odyssey was calculated when we prepared our Care for Rare application. We had an economist who calculated in Ottawa, anyways, it could cost as much as $23,000 for this so-called diagnostic Odyssey. And there's also the associated socioeconomic quality of life, sort of indirect costs to not knowing the diagnosis of a rare disease. And this really has changed with the advent of clinical genomic sequencing. The New England Journal just a couple of weeks ago had out of Baylor the clinical exome sequence experience, which mirrored essentially the Forge experience. But basically now, if you're going into a clinical genetics, a genetics clinic, it's a little unusual not to be getting your exome sequenced. And that's true in Ottawa. I'm not sure if it's true in Halifax yet, but it will be. And I think ultimately over the next decade, it'll be increasingly taken up for all of us. It will be a medical test, driven as much by initially by fad as by real need for people undergoing frontline or top-of-the-line medical care, and how we sort of collect, annotate, analyze that data is going to be one of the central challenges over the next decade. And it really is, I think, a revolutionary time that we're living in right now. Having said all the good news about the diagnoses, there is the elephant in the room of the actual therapies. And so although there's the estimated 7,000 or so, we only have therapies now for 400. And these, as I say, are increasingly rare disorders. So the idea of a fully funded drug discovery program for any of these is really comparatively unlikely. So basically, how do we treat these disorders? And so I choose to look at rare genetic disorders as a dosage problem. And sometimes the genetic disease will result in an increased protein level, rare, but it happens. The triplication of synuclein, for example, that leads to sort of early onset Parkinson's, 3 times as much synuclein, you get the disorder, or introduction of a novel pathological function, myotonic dystrophy, where you get expanded CPG repeat. And those are usually the predominantly inherited disorders. More often, it's a reduction in the level or activity of a given protein, autosomal recessive being a self-evident one. The ones we're as interested in are the haploinsufficient ones, the ones where there's a null mutation, and then there's a wild type just sitting there, non-mutated. There's GLUT1 deficiency leading to seizures is just one example of that. There's probably about 200, 300 of such disorders. And the reason we're interested in those, I think, should come clear in the second hour of my talk. So in broad terms, what we're thinking of doing is just normalizing these pathogenic trends. Rarely there's too much of it, activity or protein. More frequently there's too little. And the rather non-Nobel winning concept that we have is let's try to correct the pathogenic excess or deficit. Essentially, let's see if this is physiologic levels of a protein level and or activity, try to, and this is completely made up, this population figures, I have no idea, it's Gaussian symmetric, but I just sort of wrote it this way. Try to get, if down here is the recessive loss of function, up here the dominant gain of function, just try to push the activity and levels back into the physiological range is what we're trying to do in my laboratory. So, as I've said, we really know increasingly the data set of what genes cause what diseases and even what genes moderate, even a monogenic disorder here, modifier genes are known as well. So that data set is very well known as we approach this problem. The data set that isn't so well known is the impact that the pharmacopoeia has on the human transcriptome and thus proteome. That every time we take a medication, and I think this is Maxillatine, which is, but I'm not sure. Anyway, because I have a variety number of these slides. It's a drug that we've shown. Sorry, it's my mother phoning to see how the talk's going. I'm giving a talk, I'll call you back, bye. Anytime you give a drug, and this is just a space-filling model of a metazoan cell, the glycoproteins on the surface, the active cytoskeleton, the kinases, the little globular kinases, and it's hard to see with the light on, but the actual size of the drug we give is this little box right here as you throw it into the soup. It's clear that this is a complex topological data set, and the idea that this thing is going to bind exclusively to one area and nowhere else is really a bit optimistic and naive. So anytime anybody tells you about this specific kinase inhibitor, the specific drug, bullshit. Basically, there is a distribution of interactions, and there's no question there's going to be one that targets in particular, but there's going to be all sorts of off-target effects that occur when you give a medication. And the one that we're interested in, if this is just DNA going to RNA being read out to protein, we're interested in either drugs that bind in this realm or the pathways that modulate this that will lead to differences in the mRNA levels and ultimately protein levels. And sorting through which drugs, which are clinically active for indication X, unexpectedly modify gene Y and can be used for treatment for disorder. It is the old saw of repurposing FDA drugs for new clinical indications, something that people talk about a ton, something which is hard, easy to talk about, hard to do. So if you break it down as far as approaches, there's the increasingly revived gene replacement. Gene therapies coming along with AAV, protein replacement, as you see in insulin for diabetes. Functional replacement, a little bit trickier to do given how complex proteins are. Cellular replacement, where you actually do stem cell therapies or some other means of cellular supplementation. And then actual for neurodegenerative diseases, as George and I know, anti-apoptotic approaches. But what we're talking about here, those are sort of the 5 standard approaches, or 5 of the many approaches. We're talking about this pharmacologic gene therapy, whereby if these are the set of all mRNAs, all genes, there's a set that actually are modulating the disease in some fashion. There are a set that actually respond to drug. And it's the intersecting set of the mRNAs and proteins which modulate disease and respond to drug that we are trying to populate in our lab and in Care for Rare. Is that conceptually? I drew this myself, this figure myself. It was my artwork. So basically, I've already gone on this. For the gain of functions, we're going to try to knock down. For the loss of functions, we're going to increase. And for the loss of functions, there are the autosomal recessive that have residual activity. And if you upregulate a given recessive disorder that has, say, 5 or 10% activity, and you can actually double that, theoretically getting up to 10 or 20% activity, one knows from studies of recessive activity, you don't need a ton of upregulation to get an uptick. So basically, theoretically, that is a good approach. Although, there is something called nonsense-mediated mRNA decay. And so one of the anxieties we have, and we're encountering a little bit in the first 6 months of our project, that even if you upregulate a given amount of mRNA which encodes a flawed protein, the cell has a very sophisticated way of identifying mutated mRNA. So as you go up with it, the cell may in some cases ramp up its nonsense-mediated decay, and you just put your foot in the gas, and at the same time it's putting your foot in the brake and breaking down the mRNA. So we're a little less enthusiastic about that approach. We're more enthusiastic about the haploinsufficient ones, where there's a null mutation and then there's a wild-type mutation, a juicy drug target where it just is making the normal protein. And so you really have to make sure that's a null mutation, it's not a gain of pathogenic function or dominant negative. And if you ensure yourself it's a null mutation, then you go after that drugs that can turn up that residual wild type. It'll be turning up both, but it won't make a difference. The wild type protein will be made in greater excess. And then the one that we're talking about here are these so-called complementing genes that were first observed by David Weatherall in Oxford, looking at sicklers that got better with persistent fetal hemoglobin. There was an observation he made, I think, back in the 50s or 60s, where the babies that had this condition called persistent fetal hemoglobin did better with hemoglobinopathies than those that didn't. And that's because the fetal hemoglobin, which usually turns off within the first 2 years of life, persists and functionally recapitulates and complements the sickle cell disease. Duchenne muscular dystrophy, utrophin, and dystrophin, mutated dystrophin, utrophin theoretically, which would be complement for Duchenne, Coincidentally, Kay Davies at Oxford and David Weatherall came up with that observation. These are some other diseases that have these so-called sort of rescuing paralogs. And the one I'm going to talk about today is spinal muscular atrophy. I have no idea how much, so I'm okay. So SMA, I think many people will know about it. It is a recessive disorder which ground zero for this disorder is the motor neuron, which dies off in the first years of life, sometimes in the first months of life in the severe type 1 SMA. It's second most common after CF, autosomal recessive disorder. Every disease has its own little sort of tagline in order for funding purposes, etc. What we say about SMA is the most common inherited cause of infant mortality. Although when you think of some of the severe thalassemias in Mediterranean populations, etc., I'm not completely sure that's the case, but it's up there in the top list. It's 1 in 11,000 incidents, a frequency of 1 in 45 carriers, which in the rarefied world of rare diseases makes it one of the more common rare diseases. It's global, Chinese, African, it's out there. And it is, I say, a specific loss of motor neurons. So this ancient slide showing skeletal muscle, these are denervated skeletal muscles where the motor neuron has been lost. And these are ones where the motor neuron preserved and is maybe slightly hypertrophic, if anything. Hence the word spinal muscular atrophy. Remember that slide, George? George and I founded a biotech company around the NAPE gene, which was the wrong gene, and we used to go around showing the slide about 20 years ago or so. Type 1 SMA, unfortunately the most common form, unfortunately because it's the most severe, and you get, say, the bell-shaped chest, prominent sort of diaphragmatic breathing. For some reason in Duchenne's, SMA, and most muscular dystrophies, the diaphragm at the bottom of the lung, of the thorax is preserved, and so most of the breathing that occurs with an SMA child is so-called diaphragmatic. This is sort of a frog leg, and sort of, you can see the low tone here. There is atrophy occurring in this infant, although you can't see it with the subcutaneous fat. And this boy would die within the first 6 months of 6 to 12 months of life in the old days. These days, with aggressive respiratory intervention, they're living longer, but there's still no therapy for SMA. Here's type 2 SMA with, you can see the atrophy more clearly, kyphoscoliosis, contractures, which this is from Vic Dubowitz's book, they're no longer contractures with active occupational and physiotherapy for these kids. And you can see they're bright kids. They are neurologically intact. And because they spend their early years, formative years in a wheelchair observing their siblings racing around, they're sort of old, they're wise before their times. It's quite a poignant phenotype. They're sweet kids who sort of have a certain wisdom and a good quality of life. There doesn't seem to be a huge self-pity in their makeup. So this is the genetic architecture of the region of chromosome 5q where the genes were located. This is the NAPE gene, the neural apoptosis inhibitor protein, which we identified, which is an interesting anti-apoptotic protein, which has a whole story in and of itself. But this is the gene that causes the disease, SMN1. Because of a genomic accident, there is also SMN2. This is a hairpin duplication. And this SMN2 persists exclusively in us. No other non-human primates have it, no other species has it. So this SMN2 is a paralog which is unique to humans. And so in SMA, the SMN1 is gone totally. SMN2 persists. It's a polymorphic gene. It can exist from 0 to 6 copies of the genome. I have no copies myself, which beats the baldness of years. But anyways, and so when you look at children with SMA, generally speaking, it's a terrible x-axis here, but less severe as you go type 1, type 2, type 3. The less severe, the greater the number of copies, 3 to 4 copies. And here you see 1, 2, or 3 copies. So by and large, it's a natural experiment. They're all genetically identical. They all, in the sense, they all are missing SMN1. But as you get increasing SMN2, you get a mild disorder. So clearly, the drug target from our perspective is, can we make SMN2 sit up and fly right, make more RNA, make more protein? The deal is it's got a point mutation on exon 7, and so 80 or 90% has exon 7 deleted, only 10 to 20% is the full length RNA. But nonetheless, there's a woman in Italy that has 7 copies of SMN2, no SMN1. So genetically she's got SMA, but phenotypically completely normal. So it really is a very potent modifying gene. And so what do we do? Basically, there are microarray technology, which I don't need to tell people about here, I don't think. But anyway, so big pharma and academia, the Broad Institutes of the world, have been throwing various drugs and interventions, perturbagens, on cell lines and doing AFI arrays and to classify response, etc. There's huge data sets which exist out there. And so what we're interested in is SMN2. What happened to SMN2 in the presence of various drugs? So what we do, this is about 4 years ago, we went to the Johnson & Johnson genomic database down in La Jolla, California, and said, you know, it's great, you've got all these data sets, all these points, but could you just target this one point SMN here and tell us, there's only one address, it's SMN for SMN1 and SMN2, they're that similar. Tell us what turns that up or down. That's the only thing we're interested in. It's sort of a, are you going to eat that school of doing research that basically you just go into your data sets and find out what turns it up and down. And within 2 weeks, this guy who's a bit of my hero, Zhujin Liu, came back and showed that when they gave university students LPS, and I don't know if they still can do this these days. I mean, they're just university students, but nonetheless. And they gave LPS, SMN went up, this is a logarithmic scale, and when they gave the students an inhibitor of P38 kinase, which they were studying, it was totally gone. The induction was totally gone. So within 2 weeks, I got a report back saying SMN1 and SMN2 regulation may be downstream of the P38 pathway. So bang, that was observation 1. P38 is a stress kinase, one of the 518 kinases. It's environmental stresses, growth factors, inflammatory cytokines can lead to its induction. Here it is right here. And it leads to all sorts of transcriptional responses down there. And so I then went to my friend Justin Lamb, who worked at the Broad Institute, no longer there, who has something called a connectivity map, and asked him what happens with SMN. And he came back. They'd grown cell lines treated with 1,600 mostly clinically used drugs, 10 micromolar, a bit high, extracted the RNA 6 hours later, and did microarray analysis. And he too had a huge data set of every gene in the transcriptome, its response to 1,600 drugs. And I said, that's great, just get back to me with SMN, what goes in SMN. And he got back with the top 10, he ranks the whole drug list for us, but we just took the top 10. And lo and behold, the top 4 of the top 5 were at what were known as P38 activators. So we have Eugene Lee saying that P38 inhibition, you lose induction, and then Justin saying with P38 activation, you've got increased SMN levels. And so without having to sort of hold a pipette, we identified in silico the P38 pathway as being important in modulating SMN, which I think is increasingly the way a lot of research will be done in the years to come. There's just such a wealth of information. We just need to learn how to annotate it and access it effectively. And then moving more locally, this is Faraz Farouk. This is his Bollywood picture. He's aged a lot since then, but he's asked me to keep this picture in. And then Jeremiah Hadlin. He's now a research scientist at the Institute. He's an MD, PhD student working in my lab. And they, primarily Faraz, showed that anisomycin, which is #1 on Justin's list and one of the big activators of P38, indeed activates P38 and as well induces SMN quite readily. And when you inhibit P38, you lose the induction of SMN. So he showed in vitro that works in patient cell lines. The model, and this is my son Duncan, who's in the audience, and Faraz, who showed the in vitro model is one where HUR binding RNA protein, HUR, which is an mRNA binding protein, comes out of the nucleus, binds to SMN RNA, stabilizes it, makes more protein. So that they figured out the mechanism. It's very nice, but won't, and that's about a year's or more work, but not the main point. What Faraz then did was looked in the literature. The problem with anisomycin, it does not cross the blood-brain barrier. Celecoxib, the COX-2 inhibitor, does cross the blood-brain barrier and also activates P38. So they moved on rapidly to see if celecoxib would work the way niacin does. It does activate P38 as predicted, and there's also an increase of SMN that we see there with celecoxib. So this looks like, in vitro at least, effective repurposing. And then we go on to the mouse models. You induce SMN quite dramatically, both in the brain and the spinal cord. So there are genetically and phenotypically faithful mouse models made by Arthur Burgess in Ohio State University, among other places. They die at about 14 days. And they don't have much SMN at all when you look in the brain or in the spinal cord. And then you induce with celecoxib, very low dose of celecoxib, you get the induction. And it's hard to see with the lights on, but the green here shows it, you know, brain and spinal cord, but it could be astrocyte or glial cell or somewhere, but it really is the motor neuron where it goes up. And indeed, when you look at, there's an extension from around 14 days up to around 20 days in the survival of these mice. And in point of fact, we're a bit disappointed with that survival. We think it should be greater, and there's a reason for that I'll just get in the very end of my talk. So there's induced in motor neurons, and there's an extension of life when you give the celecoxib to the mice. Daily injections throughout every entire lifespan of the mouse. Duncan can tell you, I don't know much about it, but he can tell you the details there. There's another way of looking at inducing genes, though, in addition to doing the drug screen that we did. This is Faraz's work. Looking at the promoter, one saw that whereas the SMN1 promoter has a whole bunch of transcription factors, there's only one for SMN2, STAT5A. Prolactin, discovered by Henry Friesen, who was recently in town, I understand, is a profound STAT5 inducer. And so when they went and looked again at SMA mice, you got an even greater induction of the SMN when you gave prolactin, up to 50-fold in the brain, 8-fold in the spinal cord, and then once again, it occurs in the motor neurons specifically. And the interesting point here is that this is a heterozygote mouse. It has one SMN missing and one SMN present, 50% SMN. Prolactin gives even greater levels of SMN than it does that. And these mice live a normal lifespan. With 50% SMN, you can live normally. So you get a profound induction with prolactin, the most profound of any agent known. And yet, the longevity, it goes up above 20 days, but it doesn't go months the way we hope it would. I just want to, do I have time for a quick video to show? They're suitable for families. It's not that kind of video. So gone. Sorry, did we transfer it onto the desktop?
Yeah.
Okay. Are there any personal files I should avoid here, Sue?
No, we're good. This is, yeah.
Okay, great, here they are. Okay, here is a mouse that is given, that is dying at 14 days of age with SMA, where basically it's the end of their life, as you can see there. It's a pretty poignant video, but so they live about 13 or 14 days and then they expire. And then, on a happier note, these are the ones that were given prolactin at 21 days, a full week older. I hope this is going to work. I'll just have to do an interpretive dance of the mouse up here. Well, I guess it's not going to. At any rate, it shows the mice rather vigorously moving about the cage. And I guess the point I was trying to get, it may come on later, but the point I was trying to show is that these mice are actually dying of cardiac failure. It's not really a SMA. When you do a postmortem on the mice, the heart has profound hypertrophy and myopathic changes. And the ECG studies they show that fundamentally that's how they're dying. So we think that if we got these into humans, these drugs, celecoxib and particularly also prolactin, we would have a more profound effect than we are having in the mouse models. So our path forward with the SMA, is going to do a celecoxib trial with Lawrence Cornblit out in Calgary, Craig Campbell in London, and Hugh McMillan at our place, trying to get GMP prolactin formulated, and so that we can do a late phase 1 trial there. And then the most important thing, perhaps, is newborn screening. By and large, by the time a mother, and it's usually the mother recognizes the baby isn't moving the way he or she should, many of the motor neurons are gone. And so this is an example, as with any number of other diseases, where you really need to get in pre-symptomatically to have an effect. It's a bit sad, or that most of the fundraising is done by families who have kids who are not going to benefit from the discoveries that are being made around the world. And it's only when we get newborn tests in and make the diagnosis within the first week to 2 weeks of life, if you start the medication within the first month, that will have a really profound impact. And so the newborn screen is there technically. All we need to do now is convince the regulatory authorities that we have an effective medication. But if we can get prolactin into a baby with type 1 SMA at 1 or 2 weeks of age and show that baby sitting at 1 year, that would be an N of 1 trial that would be fairly compelling, I would think. So this has been used as a model to carry on with this Forge successor called Enhanced Care for Rare. It's headed up by the inimitable Kim Boycott, who's there in the middle, and I think has been a visitor here, as well as Jacek Nowicki, who's one of the best genome sequencers in the world for my money, as well as Deborah Marshall, an economist from Calgary, and Matt Pletcher, who's based in Cambridge, Massachusetts at Pfizer. And in addition to sequencing another 200 new molecular diagnoses, the way they did in Forge, we're going to be looking at 100 disorders in the way we looked for SMN to identify those FDA-approved drugs that can actually modulate the disease genes and just run it through a pathway where we do the in silico, in vitro, in vivo, and ultimately for 1 or 2 disorders show that this is an effective way of finding drugs. So that's pretty well it at a gallop. Basically, the conclusions are that these newly recognized disorders are subtending an increasing population of pediatric morbidity and mortality. If you walk around the wards at our hospital, because we're getting so effective at asthma, and RSV, and all that, the sort of irreducible minimum are these children with large syndromic or monogenic disorders who are, because of technology, often being saved when they wouldn't be saved before. So it's a larger portion of morbidity and mortality as we're going forward. It's very hard to see how new drugs will be fashioned for them. So translational opportunism of the sort that we describe here will be needed. There's still a challenge. These are tiny groups. And so the idea of doing phase 2, phase 3 is going to be impossible for most of them. So how one shows clinical efficacy for most of them is going to be a real challenge. New means of doing drug trials is going to be needed. And then this is what I said may work for other disorders, is all this is. I think getting corporate buy-in so that we can get the Pfizers and the Novartis to open up their data sets so we can stare at these things, I think will be an important step. And here's the group. I thought I had another one there. At any rate, this is from a couple of years ago. This is Faraz, who did a lot of the SMA work. Paco, who's the eye candy Spanish professor who visits my lab, all the girls gush about in an offensive manner, is the cell biology guy who did some of those microscopy things that we saw of the motor neuron. Jeremiah Hadwin did the mouse work. Duncan McKenzie did a lot of the mouse work as well. And I should also acknowledge, who else? David Nauman fashioned a lot of this conceptually from the start. Collaborators, Justin Lamb at the Broad, Eugene Lee and Johnson & Johnson, Lawrence in Calgary, Craig Campbell in London, Ontario. And I put Chris McMaster in Nova Scotia because we're going to have a brilliant collaboration in the future, Chris. No pressure. And this is Tori, a young lady with type 2 SMA whose family raises a lot of money for the work that's done in my laboratory. Thank you very much.
Is it mostly clinical exomes now in Ottawa, or what?
Or what's going on?
Yeah.
What's going on in Ottawa? Yeah, I mean, because I think the Boycottian effect, I think by and large, most, I mean, there could be an observer effect on my part. Most patients that I hear about that are coming through get Nexome or slated for Nexome. You're not there now? No? No? No.
But you're doing it under the
Yeah, Care for Rare.
Care for Rare,
Yeah,
not as
yeah,
a.
yeah.
I have a policy question. So you started early on and expressed a bit of skepticism about the next 5 to 10 years, and sort of wondering around whether orphan drugs and the development thereof would be subject to a lot greater scrutiny going forward.
Yeah.
Do you have any sort of blue sky thoughts about what we could do differently? What resources need to be in place? Whether it's more of the kind of public partnerships you're talking about, or if there are other models that
It's
interest
a
you?
national drug plan, and so a woman named Johanne Reeder, who is in charge of the CORD, the Canadian Organization of Rare Diseases, is having a meeting next month to sort of do a jamboree where we come up with a national disease plan, a rare disease plan. So I think that's an important step. And then the other things that need to happen, there needs to be an annotation of rare diseases and sort of standardization of how we capture them genetically and how we capture them phenotypically. And once again, Mike Brudno, working with Kim and others, is establishing this phenotypes, this means of capturing them and in a way that I think will have international uptake. So I think once we start capturing these things, get a national drug plan, and more templates, because there's so many of the rare diseases, we need standardized templates for their approach as far as natural histories, etc. I think it's going to get better, and I think we'll find it will improve. I just don't think pharma necessarily will be a dance partner.
I think one other comment that I've thought about on that front is also the price point. You know, right now there's a very few number of diseases for which there are actual effective treatments, and we all know Fabrazyme sells for $250,000 per year per patient.
Right.
If you rev that up, let's say even by tenfold the number of drugs that are out there, you can't price them all at $250,000 per year per patient. The provinces will go broke and the payers won't pay anymore.
Right, right.
And so I think the profit margin for this kind of disease is also going to decrease over time as more therapies come on, even though each disease is equally rare.
Yeah.
I think that might be driving large pharma away, and it becomes boutique biotech almost that would tackle these.
But they haven't had that thought yet. They're ahead of no trouble. But yeah, absolutely. I mean, there are cases. There's one where they did on a nocturnal sort of cryoprecipitation disease, and they did that, but they did that because they thought it would extend to rheumatoid arthritis and other things. So occasionally they'll do a rare one because of the generalized extension. But for a lot of these disorders, there is the P53 calling sort of leaf formality disease, etc. But by and large, those extensions aren't going to happen for most disorders.
And how many, have you gotten a feel for analyzing all these data sets and looking at the changes in transcription? Yeah. How many of those changes in transcription are due to on-target effects of the drug, and how many are these spurious off-target effects?
Well, I mean, who knows if they're on or off target, how reproducible they are. By and large, the problem with a lot of the AFI stuff is how frigging variable they are. So one needs to do data set correlation, where if you see it in X, you need to also see it in Y. Otherwise, you can be misled a lot of the time. They're fantastically imprecise. As more RNA-seq stuff gets in, RNA-seq is a far higher quality, more expensive, but more reliable data. So, I mean, what's on target? Everything we're looking at is probably off target. I think most of things are off target, but whether or not it's a real biological thing, a reproducible observation is the real critical question.
That's good. Joanne and Andrew.
Oh, they're lining up.
George.
It's great.
Do you know if there are other more common neurodegenerative diseases that might have a common pathway to SMA1, where this might be tested to see if it might improve?
Yeah, but ALS, they see if you have.
Because that would interest the pharma.
Yeah. So there's a P of 0.05, a very modest effect of SMN1 copy number for sporadic ALS. And surprisingly, if you have too little SMN, you get an increased ALS. And also, if you have too much SMN, there's like the sweet spot, you can also increase ALS. The effect is not so profound as if you build a drug around a toe. So we've always thought other motor neuron disorders, etc. Why SMA happens, he says, answering a question not asked, is that
I was wondering.
there's a peripheral transcriptome. The neuromuscular junction is architecturally an incredibly complex entity. There needs to be a pool of mRNAs down there to supply the material to maintain that functionally. SMN is the chief governor of that. It's an SMN, it's an mRNA transport molecule, and it's responsible, among many other things, in keeping the peripheral transcriptome up. And when you have deficient peripheral transcriptome, your neuromuscular junction doesn't work as well.
So we work in conjunction with something else.
Yes, absolutely.
Andrew.
You do this very hard.
I point and speak. Yeah.
So Alex, I noted from the tone of the first part of your talk that you're pretty enthusiastic about the potential of exomes to reveal
Yes.
disease-causing variants. Our experience has been that unless it's recessive,
It sucks.
It's really, really, really hard. And doing a genome may give you more data without much more in the way of insights.
Yeah.
Obviously, Forage's original data set included the most tractable
Yes,
diseases.
absolutely.
And I think everybody's experience has been that that's
Yeah.
not. So are you more tempered in your optimism, or are you
No,
just...
I refuse to see reality, and I'm going to carry on my antipsychotic medication. Yeah, absolutely. I mean, there's a bit of a disconnect. Theoretically, there's 3,500 or 3,000 maybe now disorders left. They've smoked through Smith's textbook. Most of those have been nailed right now. And so already we're into, it could be tougher slogging for the remaining couple of thousand, that basically there are not low-hanging fruit at all. So it's going to be tougher, but I think we're going to get better at whole genome sequencing, and also I think transcriptional mapping, whereby you take a relevant tissue and do RNA-seq or AFI and identify. We were talking about that very last night. Identify sort of anomalies in transcription. So it won't drop out. And so there is a bit of, we live in a technophilic society, and this is an instant solution to everything, but it'll get trickier than we think. Be trickier than we think, I grant you that. But I think still within the next 5 to 10 years, I think we'll have it sorted through. I guess the whole question is a non-coding question. What proportion are going to be non-coding mutations?
George, you still have your question?
Yeah.
Remember what it is?
Yeah.
Amazing.
Thanks, Chris.
None of us are getting any younger, George.
Yeah.
I'll try to
It shows. Camphor did quite well, 1.69 in terms of
Did you?
reduction. Yeah. Have you ever thought about camphor?
And so what is camphor?
Camphor is a flavonoid that
This guy's amazing.
yeah, David Kaplan has been working on it for the lupus erythropecia.
Alopecia.
Yeah, so you're joking about SMN and hair loss, but it does have a variety of growth promoting effects, mainly through cyclic AMP, but
Yeah.
interestingly enough through stress kinases as well. And the nice thing about camphor is it's very safe.
Oh, yeah. And so as we're going through and looking at all these pathways, what shows up? Genistein, camphorol,
Yeah.
lutein,
Luteolin.
luteolin, all these isoflavones or whatever,
Yeah,
these
yeah,
phytoestrogens.
the it's an isoflavone, that's a flavor. But anyway,
Okay,
yeah,
sorry, not
but they're similar.
Okay,
But diazin is an estrogenic
okay.
flavor. The
Okay.
other ones are different in terms of it, but it does suggest that there's an estrogenic effect.
Correct, there's,
There's a trophic signaling pathway that SMN seems to be connected to.
yes.
So,
Okay.
so any ideas as to what
No, I'm just, everything you say I'm writing down.
Joanne alluded to that with the synapse? Yeah, okay.
Okay.
We were talking about that. And then lastly, the COX-2. Have you
Yeah.
tried crossing the SMN knockouts with the COX-2 knockouts
No.
or anything like to see if it is
Ah,
COX-2 in fact?
Right.
Because you suggest the low dose.
what does a COX-2 in all mouse look like?
They're not so great, but
Okay.
And they're infertile.
Are they?
Yeah.
Okay. Okay.
But the dose was so low for celecox that,
Yeah, I know,
quite
I know.
get that.
Yeah, I know. Neither did the reviewers of the paper, but yeah, it's surprisingly low. We go up, we lose it. Hmm.
Okay, I'm done.
Okay,
I can't remember anything else.
there we go.
More questions from the audience? I got one, it's another log. So ERDRC is this International Rare Disease Consortium that's sort of, Paul Lasko is the head of the Institute of Genetics for CIHR now, he's the president of that for the next couple of years. What they're trying to deliver, as as you know, is 200 new therapies by 2020.
Yeah.
You think they'll get there?
It depends on semantics and what they define as new therapies. It's going to be tight. It's going to be tight. I saw one recently, and it's showing that they're off target at this stage right now. So I'm not sure. I mean, basically, $12 billion is going in here, for example. We hope to get maybe 2 or 3 new therapies at the end of the 4-year run. And so depends on the global activity, but I think they'll be lucky to hit it. I think it's unlikely they'll surpass it for sure.
I agree.
Mm-hmm. Yeah, you know from Ignite.
Yeah, it's hard. Well, you know, it's a long path to even a
Right.
preclinical path. It's long. Orphan diseases have probably the advantage is some of the shortest preclinical paths go there because there's a high level of need and patients are willing to and are willing to take a higher level of risk because of that.
Mhm.
But and the clinical trials are usually small just because they have to be.
Yeah.
So to get there is faster and cheaper, but the amount of people working on it, like you said, is still relatively small, and Big Pharma's not throwing tons of money at it yet.
Not yet, no. It could change, but yeah.
Yeah. I think the other thing, patient ascertainment and patient phenotyping, when you start one of these trials, I guess you've got to wait.
They're good at working the phones, and if they get the genetic data set, Genetic Alliance with Sharon Vance just came out yesterday, I heard, with a means of actually putting your genotype in to get into a data set. So if you start getting the annotated data sets, you can have rapid access to patients in North America and elsewhere, I think. So the idea of doing a trial, there's somebody in Belgium, somebody in Alberta, and somebody in Pennsylvania, will be tricky, but finding the patients may be getting easier.
Alrighty, being on time, I'd like to thank Alex, of course, you guys for coming.